Hello friends, dear learners and my dear student. First of all, I take an opportunity to welcome you at my T.Y.B.Sc Analytical Chemistry class. I am Dr.S.R.Labhade, working as Associate Professor in the Department of Chemistry at KT College, Nashik. Student, in previous lecture we discussed the analytical principle of flame atomic emission spectroscopy. Furthermore, we discussed the emission spectra. How does it occur? We also discussed the resonance line on the emission spectra and application of resonance line in determination of different elements which are atomized by using a flame temperature. In addition to this, we also solved some problem based on calculation of wavelength of emission as well as determination of energy of excitation. Student, in today's lecture we discuss the factor affecting the intensity of the resonance line or emission line. Student, in today's lecture we discuss the factors that affect the intensity of resonance line or emission line. The factor those affect the intensity of resonance line, that will be totally depends upon the flame temperature and number of atoms given in the excited state. Therefore, the intensity of emission or resonance line is directly proportional to the fraction of ion, the fraction of atoms that undergo excitation in the flame. The excitation or the number of atoms given in the excited state, that also depends upon the flame temperature and given by Boltzmann's equation. Therefore, Boltzmann's equation gives the fraction of ion those are excited. Therefore, number of atoms that undergo excitation upon number of atoms formed in the ground state level is equal to delta E raised to delta E upon K into T. This equation is called as Boltzmann's equation. In this case, N asterisk or N star upon N is equal to fraction of atoms that undergo excitation. In this ratio, N star, that is a number of atoms undergo excitation. Here, this N is a number of atoms formed and initially present in ground state level. Here, delta E is the statistical constant that is related with energy levels of the atoms, that is electronic energy levels of the atom. This delta E, that is the energy difference between excited state and ground state level of the atoms. Here K is called as Boltzmann's constant, and its value is equal to 1.38 into 10 raised to minus 16 Earths per degree, and this K is absolute temperature. T is absolute temperature in Kelvin. Therefore, here intensity of emission line or resonance line in atomic emission spectroscopy totally depends upon the fraction of atoms that undergo electronic excitation. Fractions of atoms that undergo electronic excitation means number of atoms those are formed in the flame. Out of that, how much are excited? Therefore, this is a ratio, that is L star upon N. L star is number of atoms that undergo electronic excitation. N is number of atoms formed in the atomization process. Delta is a statistical constant. This delta E is the energy difference between two orbitals, that is excited state as well as a ground state. K is called as Boltzmann's constant. Its value is 1.38 into 10 raised to minus 16 Earth per degree, and here T is absolute temperature. Therefore, fraction of ions those undergo excitation in the flame that determines the flame intensity or emission intensity of a resonance line. No doubt, fractions of ion those are undergo excitation also depends upon different factors. Number one, that is a flame temperature. Flame temperature that directly affect the atomization process. Therefore, here, if we have a hottest flame, then atomization process is completed, and therefore maximum amount of ions are given in the excited state. Therefore, atomization process depends upon the flame temperature, and therefore flame temperature directly affect the intensity of resonance line or emission line. Sometimes if temperature is very large, then sometimes very high temperature flame also ionizes the sample. That means without atomization, that atom are converted into ions, and when ions are formed, ions does not undergo the electronic excitation. Therefore, a fuel rich flame is used. At that time, temperature of the flame is more, and then many times rather than atomization, ionization process takes place, and that gives ions, and ions does not undergo electronic transition, and therefore that directly affect the intensity of a emission line. Next one is probability of energy available for excitation. Here in this case, the atomic orbitals have a specific energy gap, and flame must provide that much energy to excite that electron from ground state level to excited state level. And therefore here, probability of energy that directly determines the excitation of the electron from its ground state level to excited state level. If flame is unable to supply that much amount of energy, then atom does not undergo electronic excitation. If energy is very high but that is not equal to the energy difference between the two levels, then also the atom cannot undergo the electronic excitation. Therefore, flame must provide only that amount of energy which is equal to delta E. Therefore, energy difference between the ground state level and excited state level, only that amount of energy should be available from the flame to excite that particular electron from a ground state level to excited state level. Similarly here, next factor that determines the intensity of the resonance line is the probability of number of atoms undergo de-excitation. If flame supply energy atom undergo excitation, but before emission, when those atoms are get lost in the atmosphere, then also emission intensity also decreases. That means here atomization, excitation and de-excitation, total 3 steps must be carried out by that atom in the flame. Then only we can maintain the flame temperature. Then only we can maintain the emission intensity of resonance line. Therefore, probability of number of ions that undergo de-excitation also determines the emission intensity of resonance line. Next factor is a self-absorption. When the concentration of metal ion in the solution is more, at that time when emission line given by excited state atom, that is absorbed by the identical atoms, those are present in the ground state level. That means emission shown by excited atom, that emission line or that radiant power is get absorbed by similar type of atoms present in the flame, that is called as self-absorption. Therefore, self-absorption directly decreases the intensity of emission line. That means particular atom absorbs its own resonance line, and here intensity of resonance line is decreases. Generally, it will be occurs at higher concentration of the substance or a sample. And these are the different factors that determines the fraction of ion that undergo electronic transition, and that determines the emission intensity of the resonance line. Student, after discussion about the factors affecting the intensity of emission line, now we discuss the instrumentation of flame atomic emission spectroscopy. Flame atomic emission spectroscopy is a branch of atomic spectroscopy. In atomic spectroscopy, the sample nature should be atoms. All atomic spectroscopic methods are performed by a sample which are in gaseous state atoms. Therefore, here first requirement for atomic spectroscopic method is to convert the sample solution into gaseous atoms. Student, in atomic spectroscopy, when we irradiate atomic species with electromagnetic radiation, and when electromagnetic radiations are absorbed by the sample, then we can measure the intensity of absorbance or intensity of the radiant power that is absorbed at that wavelength for qualitative and quantitative analysis. In addition to this, when we irradiate some atomic species either with electromagnetic radiation or by using a thermal energy, then if these species emits a radiation, then we can also isolate this radiation or radiant power for measurement of the concentration of substance. Here, in previous lecture, we discussed the optical components. Those are required for construction of any spectroscopic instrument. And those optical components are, number one is a radiation source, and wavelength selector. Number 3 is a sample set. Number 4 is detector, and number 5 is a recorder. Therefore, these are the 5 essential components for construction of any optical spectroscopic instrument. Here, in previous lecture, we also discussed the arrangement of these optical components in a different types of instrument. Flame emission spectroscopy is a branch of flame emission spectroscopy is a branch of atomic spectroscopy in which the atoms in which sample is atomized, in which sample is atomized and excited by thermal energy and involves the measurement of radiant power emitted at a specific wavelength. This is a definition of a flame emission spectroscopy or flame atomic emission spectroscopy. In flame atomic emission spectroscopy, sample is atomized and excited by a thermal energy, and this thermal energy is provided by the flame, and it was the measurement of emission of the atoms when those are de-excited into the flame. Therefore, here in this case, first requirement is to convert that sample into atomic species, and therefore here there is requirement of a flame, that is a nebulizer and burner system, that is called as atomizer. A second component for construction of this flame emission spectrophotometer is a spectrophotometer, that is a components consisting of monochromator, detector and recorder. Therefore, here for construction of instruments of FES, we require number 1 a device to convert the sample into atom, that is called as atomizer, and second thing is to measure the emission of that particular radiation, that is requirement of spectrophotometer. Therefore, these two components are coupled for construction of the instrument, that is called as flame atomic emission spectrophotometer. Therefore, in flame atomic emission spectrophotometer, first component is the nebulizer and burner system. This is also called as atomizer. That device takes a sample solution and converts that sample into gaseous atom. Therefore, here it converts the sample into gaseous atom, therefore it is called as atomizer. Second component is a spectrophotometer. This component is required to isolate emission line and measure its intensity. Therefore, here if we consider the instrumentation flame emission spectroscopy with the molecular absorption spectroscopy, then we can replace the sample compartment with the flame, then we have a flame spectrophotometer. Now in this case, in flame atomic emission spectroscopy, flame itself acts as a radiation source. Therefore, flame is a radiation source as well as it acts as a sample cell. Sample cell. Therefore, two components, that is the radiation source and sample cell, those are performed by, those two components are nothing but the flame. That means flame itself acts as a radiation source in FES, flame emission spectroscopy, as well as it holds the sample, therefore it acts as a sample cell. Therefore, radiation which are emitted by the flame, those are isolated by a suitable monochromatic system. Therefore, here spectrophotometric components are the wavelength selector or a monochromator. Second one is a detector, and third one is the recorder. Therefore, when we arrange these 5 components in this case, then we have number 1 is a radiation source, then we have a sample cell, therefore second is a sample cell, third component is a wavelength selector or monochromator, fourth component is detector, and fifth component is recorder. Here detector, that is a photo detector, and recorder is a galvanometer which measures the current. Therefore, here these two components that are provided by a atomizer or a flame. Therefore, flame atomizer acts as a sample cell as well as a radiation source, and these 3 components are given by the spectrophotometer. Therefore, when we couple these 3 components with the flame atomizer, then we have a instrumentation of the instrument of flame atomic emission spectrophotometer. Now we draw the block diagram of this instrument, and then we discuss its instrumentation in detail. Therefore, here there is a atomizer and burner system. Therefore, to vaporize the sample as well as to atomize the sample, there is requirement of fuel gas and oxidant. Then here this is called as atomizer. From atomizer, sample is given onto the flame. Then it is called as a flame, that is a sample cell. Sample cell. Then here there is a monochromator. Function of monochromator is to isolate a particular emission line, not to obtain a monochromatic line. Function of monochromator in this flame emission spectroscopy is to isolate a resonance line emitted by the test metal present in the flame. This monochromator isolates that particular resonance line. Its intensity is detected by a photodetector. Photodetector means this device converts that radiant energy into electrical energy, and here this signal is then given to the galvanometer, or you can say a recorder. Therefore, galvanometer can be used as a recorder in the FES. Here this is a inlet for fuel gas. Then from this air is passed, and this is a capillary through which a sample solution is taken in the mixing chamber or into the atomizer, and here part of the sample is given out to the drain. This is the atomizer or mixing chamber of the atomizer. This is a block diagram of plane emission spectrophotometer. Therefore, it is a block diagram, plane atomic emission spectrophotometer. Therefore, the arrangement of components are like this. Here, atomizer that is not in the axis of plane monochromator or a detector. Therefore, here atomizer sample, actually sample is atomized into the plane. Before that, some treatment is given to the sample in this device. Then sample is given into the flame. After that, here flame evaporate the solvent, convert those atoms into gaseous atoms, and after that sample into gaseous atom. After that, it undergoes electronic excitation as well as emission. All these three events are occur in the flame. The emitted line is isolated by monochromator. Function of monochromator is to isolate that particular line, and after that the intensity of that particular line is measured by the photodetector. Sometimes a photomultiplier tube is used as a detector in this case, and galvanometer or any electrical circuit that can be used as a recorder. Student, this is about the block diagram of a plane emission spectrophotometer. Now we discuss this instrumentation in detail in upcoming lecture. With this discussion, you can find out the answers of following question. Question number 1. Explain the factors that affect intensity of emission line, second question, write Boltzmann's equation. Write Boltzmann's equation and explain the terms involved in it. Next, draw the block diagram. Of flame atomic emission spectrophotometer and explain the rearrangement of the component. Arrangement of the components. Student, with our today's discussion, you can find out the answers of this question. We discuss in detail the instrumentation of the plane emission spectroscopy or plane atomic emission spectroscopy. The first component of the plane atomic emission spectroscopy is nebulizer and burner system. For this purpose, there is requirement of pressure regulator and flow meter for fuel, gas and oxygen. Now in this block diagram, you will observe that there is inlet for fuel gas and inlet for compressed air. Here sample is taken through this capillary in a solution form by using this compressed air, and this effect is called as Venturi effect. Here fuel gas that provides a fuel for burning of the flame. Therefore, in flame emission spectroscopy, we know that as well as it holds the sample, means it is a sample set. Therefore, good and reproducible results are obtained with a properly burning flame. And to obtain a properly burning flame, we have to adjust the flow rate of fuel gas and the flow rate of the air by using a pressure regulator and flow meter. With the help of pressure regulator, we can adjust the flow rate of fuel gas, and with the help of flow meter, we can adjust the flow of this compressed air. Flow of compressed air also determines the amount of sample taken into the solution. Therefore, first requirement for the nebulizer and burner system is the pressure regulator and fuel gas, pressure regulator and flow meter for fuel gas and oxidant. Student, atomizer or the nebulizer and burner system, the purpose of nebulizer and burner system is to take the sample solution and converts it into gaseous state atoms, and this process is called as atomization. That means atomization is a process in which the liquid sample is converted to gas-phase atoms. Therefore, atomization is a process in which the liquid sample is converted into gas phase atoms. And we know that all atomic spectroscopic techniques are performed by means of sample which is in gaseous state. Therefore, atomization is the first and important step in all spectroscopic measurement. Atomization is very important step in the atomic spectroscopic measurement. Success of any atomic spectroscopy is totally depends upon the functioning of atomizer. Atomizer that converts liquid sample into gas phase atoms, as well as the atoms are excited by the atomizer means flame, as well as it measures the emission intensity. Therefore, sensitivity of the measurement, reproducibility of the measurement totally depends upon the functioning of the atomizer. Therefore, here the success of any atomic spectroscopic method is totally depends upon the functioning of the atomizer. The success of any atomic spectroscopic method is depends upon correct functioning of the atomizer. Therefore, atomizer gives the reproducible flame, then we have a reproducible result. Therefore, here the sensitivity, efficiency, and the atomizer determines the sensitivity, precision and accuracy of the measurement. Therefore, in this case, how efficiently that atomizer works, how reproducible flame is produced or a constant burning flame is produced by that atomizer, that determines the sensitivity as well as precision and accuracy of any atomic spectroscopic measurement. Therefore, atomizer, that is the most important component in a flame or in a atomic spectroscopic method. Now here, in this case, the sample solution is taken into mixing chamber by a Venturi effect. The sample solution is taken in nebulizer by a Venturi effect. Venturi action. Venturi action means there is a fine capillary of the sample solution, and to this, around this fine capillary, a compressed gas. This is a sample solution and this is a compressed air, a high pressure air, high pressure air or oxidant. Here, this automatically serves the sample inside the chamber, and this effect is called as Venturi action or Venturi effect. Therefore, around the tip of the capillary, a flow of high pressure gas is maintained, and that automatically serves the sample inside the chamber. There is no any other provision to take the sample inside the mixing chamber or inside the atomizer. The flow rate of this air, that determines the flow rate of the sample solution. Therefore, if this flow rate is constant, then we can give the sample in a constant rate into the flame. Then we have a reproducible result. Therefore, here a pressure regulator and a flow meter is required to maintain the flow rate of fuel gas and the oxidant. Therefore, Venturi action, that takes the sample inside the chamber, and high pressure gas breaks that sample into small and fine droplets. The high pressure gas breaks the liquid sample into fine droplets, which are then transported to the flame in the form of a fog or mist. And therefore, here the functioning of this atomizer, that determines the droplet size of the sample solution. Therefore, this is the first component, that is a nebulizer and burner system. When these droplets or mist given into the flame, solvent is vaporized, then it is converted to solid residue. After that, that solid residue is converted into a gas form. Gas is get decomposed to gaseous atoms, and then atoms undergo excitation.